it puts me behind in work, and that always stresses me out. So I'm just like, why do I have to be here? Why do I have to be here? It's gonna put me behind. So.
I should put it regardless.
Yeah.
We won't say anything.
Okay. Sorry, it's just taking me a second. Spring lectures, peptides, amino acids, carbohydrates, lipids, membrane. Okay. Okay. All right. So we're gonna move on to, we go from amino acids to polypeptides and proteins, and we'll talk about enzymes. We saw a little bit of this in the lab last Thursday, which was our last lab. And so to go from amino acids to peptides, we form what are called peptide bonds, right? So these are the bonds that hold together amino acids. And if we were to describe the level of protein structure here, what level of protein structure would a peptide bond represent? Primary structure, right? Because a peptide bond holds together the specific amino acid sequence, right? So this holds together primary structure for amino acids because it holds together the specific amino acid sequence, right? And so if we think about pulling apart the different levels of protein structure, primary structure is after we pulled apart all the quaternary, tertiary, and secondary structure, you start pulling on explicit covalent bonds. And these specific covalent bonds represent a functional group. Does anyone remember what the functional group is that holds together, that a peptide bond is a part of? There's a carboxylic acid side, and what other side is there, right? So when we had an ester, right? Esters are still important, these are still around, right? That was an ester, right? We had two sides to the ester molecule. What were the two sides? So we have the R group here. This is going to be the carboxylic acid side, right? And then we have this side, which is going to be the alcohol side of an ester. So a peptide bond is going to be very similar to a functional group like this, except instead of a alcohol side, it's going to have a different functional group. What do you guys think it's going to have? What are the two terminal or termini we have for peptides and amino acids? One end of the molecule and the other end. What are the two terminals called? They're two letters. There's the N terminal and then the C terminal, right? So there's a C terminal, which is the carboxylic acid terminal, and then the N terminal, right? So what's the other functional group that you think is going to be part of an amide bond? What's N? What kind of group do you think that's going to be? What functional group? Amino terminal, right? So it's going to be an amine and a carboxylic acid, right? So we'll have a COOH plus an NH3. This is going to give us, and the specific functional group is called an amide, right? When we have a carboxylic acid side and an amide side, so basically an amine right next to a carbonyl, that's called an amide functional group. So an amide functional group will look like this, N, right there. And that can be another R group over there. Where I have the hydrogen, that could also be another R group. Those are just different mono or di-substituted amide. So this is what an amide functional group is. And this forms the functional group that forms the peptide bond. Because an amino acid has the form of something like this, where we have H2N. The nitrogen is bonded to what along the protein backbone or the amino acid backbone? Does anyone remember the name of this specific carbon? It's a Greek letter. Alpha carbon, right? So this is going to be the alpha carbon. which is then bonded to the carbonyl carbon, which is the carboxylic acid side of the molecule. So this is the carboxylic acid side, and the alpha carbon has a hydrogen bonded to it, and what else does it have bonded to it? What gives the amino acid its identity and its properties? The shape will be related to it, the specific atoms will be related to it, but what group on the amino acid gives it its properties? The R group, right? So it's going to be the R group, because this R group is going to actually tell us what specific amino acid we're dealing with, right? Everything else here is the same for every single amino acid, right? We always have the amino side, we always have an alpha carbon, and we always have a carboxylic acid side to the molecule. And so what happens when we bond different amino acids together, single peptides, is we make a polypeptide, and we take the C terminus of one amino acid and bond it to the N terminus of another amino acid, right? So to do this and to go to a polypeptide, the N terminus, bless you, is bonded to C terminus of another amino acid, which I'll just abbreviate as AA. Right, so here we see carboxylic acid. Here we see the amino side of the R group. We're going to make an amide functional group by bonding the amino acid of one, the carboxylic acid side of one amino acid to the N terminus of another amino acid. So let's look at an example of a generic dipeptide. Let you guys draw that and think about it for a second. Would you guys know it's kind of low right here? Would you prefer I just draw the dipeptide on another page? Yeah, I'll draw it a little higher up. So this will be on the next page. Hey, Jenny. All right. I see you guys still writing, so I'll give you guys a second to finish writing. Peptides and amino acids. Okay. So let's go to. So, in a dipeptide, we will have the same protein backbone for a dipeptide, no matter what the amino acid sequence is, right? The polypeptide backbone is the same no matter what. So we're going to start from the N terminal. The amino group is bonded to what carbon? Alpha carbon. And the alpha carbon is then bonded to what? The carbonyl, right? The carbonyl carbon. I'll draw this in blue since I did that on the previous page. The carbonyl carbon. And we have an R group bonded to the alpha carbon and a hydrogen. When we put two peptides together, this carbonyl is now going to be directly bonded to the N terminal of another amino acid. So now we have NH here, and this amino group is then going to be bonded to what? What type of carbon? Alpha carbon, right? So it's going to be bonded to an alpha carbon, which is then bonded to the last carbon in the structure, which will be our carbonyl. And since this is the C terminus of the side, it's not going to be bonded to any other peptides. This will either be OH or O minus, most likely O minus in a neutral pH solution. And this will be bonded to another R group. And so if we were to identify all the different parts of this polypeptide, this will be what? The carboxylic acid side, which is what we call the C terminal or C terminus. This over here is called the N-terminus. If I highlight this whole part of the chain here, that is labeled as what? How would you guys call that in a polypeptide or a protein? This is what we would call the protein backbone or the peptide backbone. These two groups right here are what? These are the R groups, right? And those tell us what about our polypeptide? So it could tell you about a lot about the function of the protein, but this tells you what specific amino acids are in your polypeptide, right? If you can identify what the R groups are, then you know exactly what the two amino acids that are there, right? What's highlighted in yellow is the same for every single peptide, whether you have 1, 2, 3, you will always have this pattern of N-terminal, alpha carbon, carbonyl, N-terminal, alpha carbon, carbonyl, and that just goes on indefinitely. This is the same pattern for every protein or polypeptide backbone. And so this protein backbone is the same pattern for every amino acid sequence, which means that if you can identify the protein, the peptide backbone, you'll be able to determine everything else you need to about it, right? So on the problem set, the first example is a peptide, a polypeptide, and you're asked to identify what is the sequence, to label all the different parts of the polypeptide. The sequence you can determine by what R groups are there. The N-terminal, C-terminal, you can determine by looking at the edges of the polypeptide. And then you're also asked to label the peptide bond, which is the one thing we haven't done here. So where is the peptide bond here? Does anyone have an idea? So the peptide bond is what functional group? What was the combination of the C and N terminals? What was that called? The combination of the carboxylic acid and the amine. Is a peptide bond. It is a peptide bond. Yeah, so what organic functional group is the peptide bond? It sounds like amine, it just replaces one letter. Amide, right? And amide, this right here, is an amide functional group. And this bond right here, that is the peptide bond. So why do we call that the peptide bond? Well, that's the exact spot that we're basically snapping two amino acids together to make a polypeptide. Okay, so I'm going to draw an example peptide on the next page, and I want you guys to identify the N-terminal, C-terminal, the specific amino acid sequence, the R groups, and the peptide backbone, and then count how many peptide molecules there are. Count how many peptide bonds there are. Okay, are there any questions on this right now? When you look at this, is it what's confusing? Maya?
There could be more than one peptide bond in a...
Yep, there can be more than one peptide bond, right? Because if we have, when we thought about carbohydrates, right? There's three, but each circle represents an amino acid. There's how many bonds holding together 3 amino acids? 2, which means there's always going to be one less bond than there are amino acids in a sequence. So if you had 25 amino acids, how many peptide bonds are there going to be? 24, right? So that's always going to be the case whenever you have, for a linear structure where they're bonded like this. If they branch off, then it gets weird, but we're not worried about that. They do that in carbohydrates, but not with peptides. Okay, so if we have this specific sequence, what's another one? Identify C, N terminals, R groups, peptide bonds, and I'll just have you guys identify that. On your guys' final exam and quiz on Thursday, I'll give you guys a sheet with all the different amino acid sequences that are there. And so you'll have a chart that looks at the different amino acids, and you're just gonna basically pairing and matching each amino acid with the peptide. So I don't need you guys to memorize all the amino acids. Just be able to look at an amino acid and identify the R group and the different components to it. Let me draw the hydrogens there, just so it doesn't throw you guys off. Here, I'll always be giving you guys protein backbones that are linear like this. All right, so where is the protein backbone? It's what's going from left to right here, starting from the N terminal all the way to the C terminal. This right here is the protein backbone. These are the bonds that form the backbone of the protein. Where's the C terminal? All the way to the right, right? This is the C terminal. And what about the N terminal? Far left, right? This is the N terminal. All right, so that's good. What about the R groups? What are the different R groups here? Before we do that, let's actually label all the alpha carbons, right? Because the alpha carbon is what the R group is attached to, right? So where's the first alpha carbon? Right next to the N terminal, right? So this is an alpha carbon. Where's the next alpha carbon? Next to what? What functional group is it next to? It's in between these two functional groups right here. So this is going to be the next alpha carbon. And the third alpha carbon is this one right here, right? So how many amino acids do we have in this sequence? 3, right? There's 3 R groups, so there's 3 amino acids. 3 alpha carbons, 3 amino acids. So this is an alpha carbon, and then this is an alpha carbon. And to each alpha carbon, we have what bonded to it that gives the amino acid its identity and properties? The amide group is bonded to it, but the amide group isn't related to the properties of the specific amino acid because the amide group is what bonds to the next, that's what forms the peptide bond, right? The properties and the identity of the amino acid are given by the R group that's bonded to the alpha carbon, right? If we look back here, we see that for a generic dipeptide here, we have two R groups that are bonded to the alpha carbon. Those give the amino acid its identities and its property. So what is the first R group we have here? It's just a hydrogen, right? So this is one R group right there. And so you say, well, that's just a hydrogen. Well, this would be the only amino acid that is not chiral. Does anyone remember what that amino acid was? This was glycine. So the R group for glycine is just a hydrogen. What about the next R group? What should I circle? You don't have to know the identity of the R group, just identifying what it is. Yeah, what functional group is on the next R group? Right, whenever I say functional group, I'm referring to the organic functional groups on the molecule, right? So where is the R group? I think you guys have an idea of where the R group is. I'm just asking a different question. I'm saying, well, what is the functional group that's on this R group? What does it look like? What atoms are involved in the functional group? Carbon, and what else? And what?
Alcohol.
So there's an alcohol there too, right? And what's the last part of that? Yeah, there's a carbonyl there, which means when you have this, so if we were to highlight this right here, that's the functional group that's on this R group. It's a carboxylic acid, which is alcohol, carbonyl, and then there's a carbon that's bonded to part of the carbonyl, right? So this whole thing right here is the R group. And this is called glutamic acid, is the identity of this amino acid. So why do you think it has acid in the name? Because it's a carboxylic acid functional group, which means this is probably an acidic R group, or one that has a chance to be charged. And what's the last R group here? Right before the C terminal. What does it look like? Does anyone remember the name of that isomer? Close. It's one less carbon. Isopropyl. Yep, that's isopropyl, right? So this is isopropyl R group. And the name of this amino acid is called valine. I'd always remember it because it looks like it has a V bonded to it as an R group, right? So V for valine. So that's valine, right? So now we have all the R groups circled in red. And you guys weren't asked to do this, but the specific amino acid, 3-letter amino acid sequence, which is what you guys are asked on your problem set to do, it would be glycine, glutamic acid, and valine. Those would be the 3-letter sequences to describe each amino acid. And so on the amino acid chart, you would be given the peptide, the single letter amino acid identifier, and the 3-letter amino acid identifier. And so I'll show you guys an example of once we finish this, of what that sheet looks like. And I think, let me see if I posted it on Canvas. I did not, so I'll post it on Canvas as well. Just so you guys have access to what you'll have access right now to what you'll have access on the quiz and the final exam. Ow. Question?
So for the vowel, they're all gonna be like that. For the R group, the name, the vowel.
Valine is the identity of the amino acid. So each amino acid is given by the R group and the alpha carbon, N terminal, C terminal. That's how each amino acid is identified. So that is valine. If it has that structure, that's valine. That next to it is glutamic acid. That's what glutamic acid looked like. That's what the specific molecule looked like. Yeah, that is like, if we had methane, we call it methane because that's what that molecule is, right? That's the identity of that molecule. That would be glutamic acid, and that would be glycine, right? So these are the same way when you have bases in DNA and RNA, where A, G, C, T, or AU, GC, those bases give the identity of the base in the DNA or RNA. These amino acids give the identity of the amino acid. These R groups give the identity of the amino acid in the peptide sequence. And so I'll go and show you guys an example of the sheet that you'll have. Do Chem 66, spring. Does this one have it? Nope. See if I can. I'll have to find it. I'll find it for you before the next one. Chem 3, lectures, peptides, enzymes. Okay, and so the last part is asking to identify the peptide bonds. The peptide bonds are what functional group? Amide, right? So those are going to be the amides. And the amides are where on this molecule? Where are the amides? Where are the peptide bonds?
Between the N and the O.
Yep, the carbonyl carbon, right? So this right here would be an amide bond. And there should be how many amide bonds for 3 amino acids? 2, right? And where's the other amide bond then? That should be right here. These are the 2 amide bonds. So that's going to be highlighted in purple. Those are the 2 amide bonds. The amide functional group is whenever we have the carbonyl next to an amino side of the molecule. Right, so this would be the first step in looking at any polypeptide. Identify the backbone, where are the alpha carbons, what's sticking out from them. That gives you the specific amino acid sequence. The peptide bonds are what hold the different amino acids together, bless you. And then the C and N terminal are the edges of the molecule. All right, any questions on that? All right, so this is the same basic structure for any protein. So protein can have hundreds to thousands of amino acids as part of them. This is looking at a microscopic view, the specific chemical structure of an amino acid sequence into proteins and peptides. All right, any questions? Right, so on the homework set, you're given this polypeptide and then the pKa value for each of the R groups, which then tell you, relative to the pH of the solution, what should the protonation state be. So for glutamic acid, this can be protonated or deprotonated. Valine, there will be no pKa because there is no acidic, you can't protonate or deprotonate that R group. It's only for the charged polar R groups that you can do that. You can also have polar uncharged R groups, and then valine would fall under the nonpolar R groups. All right. And so what forces hold together protein structure? So secondary, tertiary, and quaternary structure. Well, we have charge interactions. These will be plus and minus R groups interacting with each other. So we can have an arginine and glutamic acid. Those can interact the same way ionic compounds do, like sodium and chlorine, to form sodium chloride. We can have hydrogen bonding interaction. So hydrogen bonding occurs when what specific scenario? Under what specific scenario? Yep, if we have an H bonded to, what were the 3? Atom? Yep, F-O-N, right? And so in proteins, it's mostly going to be oxygen and nitrogen that are going to be contributing to hydrogen bonding. And so this is going to be predominantly what holds together secondary structure we talked about, because you can have hydrogen bonding between the carbonyls and the amino groups along the actual protein backbone. So if, that linear structure I drew on the previous page, the backbone, if you rotate that and bend it in three-dimensional space, then you can have the amino group and carbonyls actually interacting with each other through that way. You can get disulfide linkages because some R groups, like cysteine, have what are called thiol groups on them, or they have sulfur and hydrogen bonded to each other. So let's say that these are the rest of the protein, these are the R groups for those amino acids. These can come together to form what's called a disulfide linkage. And so those also can hold together elements of secondary and tertiary structure. So this is thiol groups covalently bonding. And those are important for metabolic process, the electron transport chain. And then we can have dispersion forces. So dispersion forces occur between what molecules? What's required for London dispersion forces? It's always present, every single molecule, right? So that's like the weakest force, but these are going to be the forces that hold together any type of molecule, right? So this is for the strongest interaction for nonpolar. And another way of saying that would be hydrophobic proteins. Right, so if we had valine and a bunch of leucine, and these nonpolar amino acid R groups, then London dispersion forces would be the strongest force that we have there. And all these different interactions give rise to a structure which is intimately related to what for proteins? Function, right? Structure and function go hand in hand in proteins. You destroy the structure, you destroy the function. For example, on your lab on last Thursday, you guys saw by adding acid or base to your enzymes what happened to the activity of catalase, which decomposed hydrogen peroxide. Was there more or less bubbles when you had it in acid or base? More bubbles? You have more bubbles in your acid and base solution for hydrogen peroxide? Well, that's not supposed to happen, but usually if you add acid or base, it should denature the protein, destroy it, and then you don't get the function of the enzyme anymore. I could have swore I saw less bubbles in those ones, but it was a long weekend ago, so. Yeah, if there was, it should destroy any protein that's there, but that's fine. All right, so these are the primary interactions. What are the different levels of structure? So we talked about this last time, but I just want to reiterate. What's the lowest level of protein structure? Primary. What's the second lowest level? Secondary. Then we have tertiary. And then lastly, quaternary. So then we have quaternary structure and then tertiary structure. And so primary structure was what again? Primary structure is, yeah, the covalent interactions, which is the specific what sequence of a protein. The sequence of what? For primary structure. Yeah, they have peptide bonds, and what do those hold together? What is the building blocks for protein? Amino acids, right? So this is the specific amino acid sequence. Secondary structure was interactions between amino acids. And is it within a chain or between two different peptide chains for secondary structure? Within a chain, right? So this should be within the same chain, right? So amino acids within the same chain. And then tertiary was interactions between two secondary structures within a chain. Between secondary structures within a chain. And then quaternary was just defined by what? So everything so far has been within a chain, right?
Oh, two chains.
Yep, quaternary structure is when we have interactions between two different peptide chains. Or two different protein chains. Peptide chains or polypeptides. And so in primary structure, what are the forces or the types of bonds that hold together primary structure? These are going to be covalent bonds. And then specifically, what was that bond called? The peptide bond? Yeah, that was the peptide bond. And what functional group was that? Amide functional group. Secondary structure is going to be held together mostly by hydrogen bonding and disulfide linkages. Tertiary structure, a lot of time is held by hydrophobic and charge interactions. And quaternary structure is almost exclusively hydrophobic interactions. The reality is that there's a little bit of mixing and matching between all of these in different levels of protein structure. Nature doesn't fit nicely into each of these categories, but these are the predominant interactions that we see in each level of structure. And so, for example, if we have an alpha helix, it'll look something like this in our cartoon drawings. These are held together oftentimes by the carbonyl of one part of the chain interacting with the N-terminal of another that then form a hydrogen bond together. So that forms a hydrogen bond. And this is happening all along the chain. Carbonyl, amino group, hydrogen bond. And so I'm not drawing the R groups here, I'm just drawing what's being hydrogen bonding along the backbone. And so for an alpha helix, which is what this structure looks like, which is going to be that spiral in the ribbon structure, it's the backbone itself that's hydrogen bonding with other parts of the backbone to then fold the structure in a specific way. The only two, and these are elements of secondary structure. The only two elements I want you guys to know for secondary structure are going to be the alpha helix and the beta sheet. There are more complicated secondary structures. So a beta sheet is going to look something like this. There's a little disordered part of the protein. In the same way that it's held together for the alpha helix, it can be held together for a beta sheet. And what these look like in the ribbon structures is they tend to look like arrows because they give it some directionality. So if I ever give you a big protein and you see these arrows in the middle of it, that's usually what a beta sheet looks like. This is what's called the ribbon structure. And then last time we talked a little bit about how you can have tertiary and secondary structures with two different alpha helixes interacting with each other. They can interact via the R groups, through polar, nonpolar, or charge interactions. If it's within the same chain, it's called tertiary structure, and if it's between two different chains, it's called tertiary structure. All right, so we'll take a break there. Let's take like a 10-minute break. We'll come back, we'll finish up proteins, and then we'll start lipids. This one? All right, so we talked about secondary structure because we didn't explicitly talk about alpha helixes and beta sheets, so I wanted to comment on that. Tertiary and quaternary structure we talked about last time in last lecture on how to identify that, right? So we can look at the destruction of protein structure. This is important because structures are intimately related to what? Function, right? Structure equals function. When we destroy protein or it loses its structure or it's changed in some way, this is called denaturation of the protein structure. And so certain things can affect this. So on Thursday, you guys saw two examples of things that can affect protein structure. What were the two things you guys tested for?
Acids.
Changing in pH. And what else? Heating it up, right? Heating up or change in temperature, I'll say that incorporates both cooling and heating up, change in temperature. And so both of these affect protein structure. So why did a change in pH affect the protein structure? Does anyone remember? What happens when you change the pH of the solution? It denatures the protein, but what specifically, what causes the denaturation? The protonation, right? So last time we talked about if you change the pH, it changes whether the side chains, the end termini, become charged or not charged, right? So this specific mechanism was changing charges on R groups. If you change the charge, all of a sudden, if you have two amino acid side chains that were hydrogen bonded to each other, like glutamic acid and aspartic acid, and all of a sudden you drop the pH so that they're both negatively charged, two negatively charged things are going to do what? Repel each other, right? So they're going to blow apart like that, where before they were hydrogen bonded. That's going to change the structure of the protein a lot when you have that happen. A change in temperature, how do you guys think that's going to, right? So when you heat something up in general, like if you heat up sugar or salt in water, what happens? Is it easier to dissolve sugar in cold or hot water? Hot water, right? Because the crystals that are held together by intermolecular forces, it's harder for them to stay bonded to each other if the temperature is high, right? So the change in temperature causes things to melt. Which is the same thing. Both of these fall under the category that whenever you're denaturing a protein, you're interfering with what thing that never goes away in chemistry? Intermolecular forces, right? So anything that interferes with intermolecular forces. For short, that's always IMF, right? So if you have anything that interferes with the intermolecular forces of a protein, that will denature it. And some other things that can do that, we can have light. I'll put it a little higher because I know that that's going to be hard to see. We can have light, heavy metals, and alcohols. Right, so alcohols, that OH group, can interfere with hydrogen bonding in proteins, causes them to denature. Heavy metals can act very acidic or basic in solution, cause protein denature. One?
What's the underlying word? Like, what's it called again?
Extra?
No.
Oh, denaturation. Yeah, that's pretty bad.
Sorry.
No, you're good. Right. Denaturation, or taking it out of its natural state. I forgot an O in alcohol. Alcohol. So if you put proteins in a very concentrated alcohol, that will mess with the protein hydrogen bonding. Light, if you make it into a radical or something, which we haven't talked about, it's out of the scope of this class, that can cause it to be very reactive, react with something else. So these are the things that'll denature a protein. Any questions on that? Did everyone really see more bubbling with the acidic and basic conditions? No? Okay. Hopefully not. If you did see it, then I'm not sure what's going on. I have to think about that for a little bit.
Two, right?
Like with the banana.
Yeah, with the banana. Yeah, if you had the banana acid and hydrogen peroxide, or if you just had water and hydrogen peroxide and banana, which one did you see more bubbling?
With hydrogen peroxide and banana has some of the.
Yeah, and then if you add acid to that or base, it should have been less bubbling.
Oh, yes.
Okay, all right. So yeah, so we denature the proteins, and then, yeah, okay, we got them. All right, good. Glad we got to the bottom of that. That was actually in the back of my mind. I'm talking up here and I'm thinking about it the whole time. Like, how'd that happen? Right, so all of those can affect the structure of the protein. And your protein specifically was an enzyme. And so an enzyme was what? I know we talked about this on Thursday, so we'll go through it kind of quick. What is an enzyme? A biological catalyst, right? It's a biological catalyst. And a catalyst is something that does what? speeds up a reaction, right? It speeds up a reaction, and it does so by doing what? There's a specific thing we talked about. It lowers something. Lowers what type of energy? Activation energy, right? This lowers the activation energy, right? And it's important for a catalyst to be regenerated so then it can lower the activation energy for another reaction, right? That was one of the other properties of a catalyst. And so a protein is known to be an enzyme if it ends with what? Does anyone know what tells us if it's a catalyst? Does anyone remember what the enzyme was called on Thursday? It is a very, I think, in-your-face kind of name for what it does. It's called catalase, right? So it's a catalyst, and the ase ending tells us that it's an enzyme. So hydrogenase, catalase, polymerase, anything that ends with ase is a catalyst or an enzyme. So polymerase, catalase, hydrogenase. All of those are enzymes. That's why they all end with the ASE ending. There are some exceptions. Pepsin, for example, is an enzyme in your stomach that digests proteins, breaks apart protein bonds. That one doesn't have an ACE ending. And so an enzyme works by lowering EA, right? And EA is activation energy. And so we can draw the energy diagrams for a catalyzed reaction involving an enzyme. And so let's do it for an endothermic reaction. So on the problem set, for example, you're given just the coordinates, or the x and y axis, which is the reaction coordinate. On the y axis, we have the energy. We'll draw both a catalyzed and uncatalyzed reaction. If I draw the products here, they can start at any arbitrary place. I'm sorry, not the products, the reactants. Where should the products be for an endothermic reaction? In an endothermic reaction, do we release heat or do we absorb heat? Absorb, right? Which means that we've taken in energy. So should the energy of the products be higher or lower than the reactants? If we're taking it in, higher, right? Because we've absorbed energy, we need to be higher in energy now, right? So the products are going to be somewhere here. And I'll draw the uncatalyzed reaction first. It would be something like this. And we can label the different quantities here. So what would the enthalpy be? Let's label three things. We'll label delta H, E sub A uncatalyzed, and then E sub A catalyzed. So what would delta H be here? It's the heat, right? And how would I label that, draw that on this plot? Yep, this is the energy of the reactants. It's the difference between the reactants and what? Delta H, the heat absorbed. Right, that tells us if it's endothermic or exothermic, right? And when we were just discussing where do I put the products, we're saying where do I put the products if it's endothermic or exothermic, right? Because they're higher in energy. So this delta H should be the difference between equals the difference in energy between what? The reactants and the products, right? Reactants and products. In how we represent that on the graph is that's going to be this height right here. Right, this will be delta H. And is that going to change if we're catalyzed or uncatalyzed? Does delta H ever change? What do you guys think? It should be the same. So why should it be the same? A catalyst works by lowering what? The activation energy, right? But you're still going from the same reactants to the same products. So those are just the same no matter what, right? The energy of the reactants and the products are the same no matter what. We can think of it as like an analogy if we are building a tunnel and climbing into a valley over a mountain, right? So if this is a mountain, there's a valley on the other side, and then there's what you're hiking over. And we cut a tunnel through the mountain. There's two paths we could take. We could climb all the way over, which is going to take a lot of energy to be able to do that, for then you to be on the other side, right? Or we could go up and through the tunnel. But the end result is still the same, right? You're still just as high up in this new valley compared to where you started from. So if we think about that in terms of reactants and products, you still start from, let's say, a water molecule over here, and you still end with a hydrogen peroxide molecule over here. The path by which you got there doesn't change where you ended up and where you started. All that it did was it just sped up how you got there, right? So if we think about the valley analogy, it's the same idea here, except instead of potential energy and gravity being in a tall valley versus in, by the Dead Sea or something like that, you're looking at the energy that's in the chemical bonds, right? So you're still starting from and ending up at the same place. So that's why the enthalpy will never change in a reaction. If we have a catalyzed reaction though, what will change? All right, if I was to draw a curve now for a catalyzed reaction, what will it look like? What will be different or the same on the current curve that I've drawn? Yep, it's gonna be the start here, we're still gonna end there, it's just we're not gonna go as high up, right? Because we've lowered the activation energy. So this curve is probably going to be something like this. We still start and end at the same place. The only difference is that we didn't have to put as much energy to go over. So this quantity right here will be the activation energy for the catalyzed reaction. And this quantity right here, will be the activation energy for the uncatalyzed reaction. Okay. And which one looks larger? The uncatalyzed one, right? Which is exactly what we said. That's how an enzyme should function, or a catalyst should function, by lowering the activation energy for the reaction. And so one of the questions on the problem set is draw two curves, one for a catalyzed reaction and one for an uncatalyzed reaction, and label these different components for it. All right, any questions on this? If it was exothermic, what changes on this plot? the products are just lower, right? We'll just make the products, they're gonna go all the way down, they're gonna go past the 0 right here, and they're gonna go down here. So delta H will then be that difference. You can keep everything else the same, and it would be identical, or it would still work, right? Because you still have to put less energy for the reaction to happen. Okay. Any questions on that? Okay, so I'm going to skip the next slide, which is talking about classification of enzymes. It wasn't something I would test you guys on anyway. There's usually a number that's associated with an enzyme in decimal places, but it isn't that important, so we're going to go straight to the enzyme substrate complex. So we talked a little bit about this during the lab too, is that the molecule that binds the substrate, or the enzyme, is called the substrate. And we talked about some of the things that determine what the substrate will be for a molecule. Usually, we draw the different complexes and talk about an induced fit or a lock and key type mechanism for a substrate and an enzyme. And so the binding of a substrate to an enzyme is determined by intermolecular forces, these are going to be determined by intermolecular forces. So, for example, if we have an enzyme that has an active site, where this is called the active site for an enzyme, that has a lot of positively charged protein R groups in that active site, the substrate will probably be what? What can you determine about the properties of the substrate? If there's a lot of positive charges in here. So let's say there's a lot of positive charges. What do you think the substrate will have all over it? Negative charges, right? So it'll probably have negative charges all over it because the intermolecular forces should match between the two, right? If we have a negatively charged substrate, then we should have a positively charged active site. Another thing that'll affect the enzyme binding is going to be the shape of the active site plus the substrate. So if we have different negatively charged substrates, then maybe only one shape molecule will fit into the active site, right? So we can be selective this way. So for example, if we had another negatively charged substrate molecule, but the shape didn't match, this would not go into the active site. And so proteins and stuff are fun a lot of the time just because it's a lot of picture drawing. So make sure you brush up on your artistic skills for the final next week. And so usually what happens is we get an initial binding of the enzyme in the substrate, and then we get a conformational change or a change in the structure of the enzyme to fit the substrate. And so this is a conformational change. Change or change in shape of the enzyme upon binding to the substrate. Conformational? So let me write that. And this is what gives an enzyme specificity, is its ability to fit to a specific substrate. And so the specific reaction pathway that we talked about in lab last time was the formation of the ES complex, where we have an enzyme plus a substrate. They come together to form the enzyme substrate complex. Which then regenerates the enzyme and we get our product molecules. In this reaction pathway, can be interfered with. And this is what we're going to call different types of regulation or inhibition of the enzyme. And so we can look at certain things like the rate of reaction. How much is the reaction being sped up by the presence of an enzyme, and see how that affects we'll see how that affects the behavior of the enzyme and its ability to speed up the reaction. And so this falls under the category of the regulations of enzyme. This is called inhibition. Concentration. Once we increase the concentration of the substrate, which is represented by these brackets, to a certain point, we become saturated. And the temperature and pH, these affect regulations of enzyme because they can denature the enzymes. But we'll talk a little bit about how the concentration of the substrate can affect the enzyme activity. All right, so when we think of saturation, what do we think of? Yeah, right. So if we're talking about an organic molecule, that means that every spot that there's a hydrogen, there can be, right? When we think about enzyme saturation, if we have a certain number of active sites, once we increase the substrate such that the active site is always occupied, then we've reached the saturation point, right? If the active site is occupied 100% of the time, if you add more substrate molecules, do you think the reaction rate will go up, down, or stay the same? It should stay the same, because if it's already working as fast as it can, you have people on an assembly line. If everyone's already working as fast as you can, if you add more parts, you're not going to increase the production at all because all the hands are already occupied. And so on the next slide, I'll draw I'll draw our next picture. I'll draw a diagram or a graph of what that looks like. Okay. So if we were to plot the concentration of the substrate versus the reaction rate, and how did you guys measure reaction rate on Thursday? How did you measure that? How did you know if the reaction was happening faster or slower? Bubbles, right? More or less bubbles, right? So if you can measure the reaction rate by some indicator, and we want to see how does the enzyme speed up the reaction. Enzyme reaction rate, we call this kinetics. If we have 0 substrate molecules, what do you think the reaction rate will be? All right, so I'll draw the enzyme plus substrate goes to enzyme substrate complex to the enzyme plus product, right? So the reaction rate measures this, the whole progress of this. All right, so the reaction rate tells us how fast is this going to completion. If we have 0 substrate molecules, what do you think the rate of the reaction will be? There'd be no reaction, so the reaction rate would be 0, right? If we have 0 S, then this is going to be 0, right? So we're going to start the plot at 0. If there's 0 S, then the rate equals 0, right? Because we have no reactions to speed up, so the rate has to be 0. If we increase the substrate, now what do you think is going to happen to the reaction rate? It'll go up, down, or stay the same? It starts to go up, right? Because as we add molecules, now the reaction can happen, so the rate should go up. So it starts to go up, 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 and then we hit a point where we saturate all the active sites. All right, so we hit the saturation point, which means that making the enzyme substrate complex can't happen anymore than it already is, right? And so then you guys a second ago, you said, well, once you reach saturation, what's going to happen to the reaction rate? It'll go up, down, or stay the same. It should stay the same now because we can't speed it up any more than we already have. So no matter how much more S we add to the solution, it'll just stay the same. So we get a plateau of the reaction rate. So this right here would be the point of saturation. Bless you. So once we reach the saturation point, we're already doing this as much as we can because we're using up all of the enzyme that's available. So no matter how much more S we put in there, it's not going to push this to happen any faster. All right, any questions on how, if you were given a plot, you could identify the saturation point here? All right, so now we'll talk about the specific types of inhibition. So different types of inhibitors for enzymes. We have competitors, which means that they bind to active site. So the substrate cannot. So if we had an enzyme, then we had two molecules, we had our substrate, and we had our inhibitor, which is I. The inhibitor could bind so that the substrate no longer can bind. All right, so competitively, meaning it takes the spot of the substrate so that the substrate can no longer bind, which means the reaction cannot happen. Non-competitive inhibition regulates and stops binding in some way. And so this is usually allosteric inhibition. And what that means is allosteric, this is a conformational change. That changes active site shape. And so I'm going to draw a picture of that on the next slide once you guys are done writing. And so what that looks like is, let's say we have an enzyme where this is our active site. We have an inhibitor molecule and our usual substrate. If the inhibitor binds some other part of the enzyme, we go through an allosteric change. Where the active site has now changed shape. So the substrate can no longer bind the active site, right? So this little inhibitor binds another part of the enzyme. It causes an allosteric change in the active site of the enzyme that now the substrate can't bind, right? So the induced fit is no longer an option. It changes the shape of the active site. It could change the charge or something else too, but in general, it's easy to think of it changing the shape of the active site. And so this is how you can allosterically or non-competitively inhibit an enzyme. Is you bind some other part of it that deforms the enzyme, which then makes it so that the actual molecule you're trying to bind no longer can bind there anymore. And so this change can be reversible or irreversible. In biological pathways, this change is reversible a lot because certain signals in the cells will tell you whether or not to inhibit an enzyme or activate it. And so irreversible versus reversible inhibition just says, can you reverse this inhibition or is it a permanent change, right? If you have a heavy metal that binds an enzyme, it might never unbind and that'll be permanently inhibited. Can you reverse